I will try to give a brief overview about the genomes of monkeypox, and more importantly, what are the insights that we have gained and the surprises that we have sort of encountered. And before I start my talk, because I believe the audience is mostly clinicians, I would also want to go back to a bit of history and to also state that practically microbiology that we do is thanks to this person, Anthony von Leeuwenhoek, who was a merchant and a municipal official by profession and a scientist by hobby. Now, what he invented was essentially this handheld microscope, through which, of course, you could observe the world around that individual, and he could explain and also sort of describe a lot of organisms which were hitherto not seen by the naked eye. Now, of course, this is not what we use to identify microscopes, but to also state that traditional microbiology indeed relies on the isolation of the pathogen and observation of the pathogen. Now, meanwhile, what has really happened in the last decade or so has been a set of techniques which could sequence nucleotides at quite scalable, fast, and quite effectively in terms of cost methodologies, which could now obviate the need of traditional microbiology altogether. And we have seen that quite extensively being used in the SARS-CoV-2 pandemic. And we would also see this increasingly being used not just for monkeypox, but also for a variety of other outbreaks that would happen in the future. And the advantage being that you could have direct molecular analysis, you could have detection, additionally with genomic characterization at a much smaller time scale, much faster, and in many ways, much more cost effective. Now, when you do a genome of an organism, what you're essentially looking for is essentially mutations or changes in the nucleotides in the genome. And different genomes, of course, would have different rates of genetic changes or accumulating genetic changes. And typically, it is widely believed and also estimated from genomes that are known in public domain that DNA viruses do have a much lower rate of mutations compared to RNA viruses. Now, RNA viruses, the examples are, of course, SARS-CoV-2. And when we talk about monkeypox, it's essentially a DNA virus. So we are essentially talking about a virus which had a basal a low level of accumulating genetic mutation. And therefore, it's not surprising that in early communications, it is widely said that monkeypox doesn't really mutate much faster, and therefore very unlikely that genomics and genetic epidemiology could essentially be used for monkeypox compared to SARS-CoV-2. Now, before I come to monkeypox, to give you a brief of the basics, so essentially what we do is you could sequence or you could look at sequences back in time deposited in public domain, and essentially you're going to look at mutations and the order of mutations, because a new mutation would always occur in the background of an existing mutation. And you're essentially going to now estimate the proportions of each of these different kinds of mutations or genomes having different kinds of mutations vis-a-vis each other, and to also look at what we call as emergence events. So emergence events are essentially a much higher proportion than expected for a particular combination of genomes or mutations. And this largely could be because of two factors, the host factors, like, for example, a super spreader event, or viral factors, for example, a virus which could be more transmissible or can evade the immune response. So essentially what we are going to look for is combinations of mutations which are suggestive of emergence events. And of course, when I said that viruses could mutate and viruses could mutate at a very constant rate, now this information is very useful because then you can use these mutations to put genomes together or cluster them together and sort of see what are the set of genomes which cluster together, and these clusters of genomes which are linked is what we call as a clade or what they call as a lineage. So essentially, clade or lineage doesn't necessarily mean that it has a phenotypic outcome or a difference in the phenotypic outcome. It only means that they have shared genetic mutations and a probably common ancestor to this particular closed cluster. Now, to keep in fact that I said before that you have a constant rate of mutations, and these mutations are sort of estimated for practically all the major organisms out there because you have genomes to go back and estimate such mutations. Now, given the fact that you have a very constant rate of mutations, and this is because of the regular processes in biology which involve base editing and base repair, you could actually use this information to actually backtrack. Given a set of genomes, you could backtrack to create a virtual organism which is sort of an ancestor for that particular cluster. And since you know the rate of mutations, you could also estimate what is in time did this ancestor arise, which is called the time to the most recent common ancestor, or in short, TMRCA. So in other words, given a cluster of genomes, you can actually go back and ask this question: when did this particular cluster arise, and what point in time did this particular cluster arise? Now, of course, Dr. Himanshu has alluded to this, so I'll not explain much in detail. The initial cluster was from 18th of May 2022 from Spain, and of course, by July 23, it was declared as a public health or an epidemic of public health importance. There are multiple cases now, close to 30,000 pod cases. Now, if you go back to the genome, of course, it's a DNA virus with a much larger genome, around 200,000 base pairs. And very important thing is that it has a very sort of central conserved core with a set of genes in the middle, which are quite conserved across pox viruses, not just orthopox viruses, but across the pox viruses, many of them which infect animals. And of course, a quite variable region on both sides, and a repeat region on the two different extremes. And the genomes of pox viruses sort of suggest that it is a very low rate of mutation, as I sort of suggested before, being a DNA virus. And this is estimated to be approximately 1 to 2 substitutions per genome per year. And that's a really, really small number of genetic mutations that a typical pox virus can accumulate over years. Now, the first surprise is, of course, the initial genomes that came out of multiple different countries. And this was available as a preprint earlier and now published in Nature. It sort of suggested that there are around 50 genetic changes compared to the viruses isolated from Africa and travelers out of Africa in 2018, 2019. And this is really, really surprising, because with a mutation rate which is expected of 1 to 2 mutations, you would hardly expect 4 to 5 mutations to have happened, but there's a whooping large number of mutations that have happened in between. And a significant large majority of these mutations were of the GA to AA or TC to TT type, which sort of suggests that there are specific molecular mechanisms involved in this particular mutations. And this particular type of mutation is suggestive of a particular set of enzymes in the human body called ApoBec3, to be specific, ApoBec3A, which essentially means that the virus is probably replicating in the cytoplasm of the cell, and these mutations are introduced by the ApoBec3 class of enzymes. Now, so that's also very surprising, because ApoBec3 and its action has never been described in pox viruses. And more importantly, this is a new information, because monkeypox has largely been a zoonotic disease where you have infections from out of animals with limited human-to-human spread. And this is probably one of the largest and known cluster of human-to-human spread for monkeypox out there. And of course, the paper also describes the cluster, and the cluster sort of suggests that a significant large majority of the genomes do belong to one single cluster, which we call as the B.1 cluster, which is actually a subgroup of the clade 3. Now, to put in perspective, of course, we don't use geographical terms now, but of course, for people who have been following monkeypox genomes out there, the earlier classification involved terms like the West African or the Central African or the Congo Basin clades. We don't, and we should not probably use such nomenclature. So the clade 2 and the clade 3 are essentially what we call as the West African in the nomenclature used earlier. In the paper, they also show these 3 genomes out here, which is called the A3 lineage. But of course, there is not much of mention of this particular lineage, but significant large majority of the genomes fell into this one single cluster, which shows this property called polyploidy or epiploidy, which essentially means that they're very closely related to each other and probably suggesting a super spreader event. Now, that's very important, because the structure of the cluster could actually sort of give insights into how this was transmitted. And very large number of genomes close together and showing this particular pattern would suggest a super spreader event. And epidemiologically, that's what we know, that this was largely a super spreader event originating from Spain, and then, of course, smaller super spreader events across entire Europe. Now, of course, thanks to the team at the National Institute of Virology, we have heard Dr. Pragya talk about it. The 2 genomes of India did come in public domain, and that is really interesting, because they do cluster not to the B1 cluster, which is associated with the outbreak event, but to a much smaller cluster, what they call as the A2 cluster. Now, the A2 cluster is largely composed of genomes out of India, from the United States of America, and from Thailand. you could sort of time the origin of the ancestor, and that is sometime around July of 2021. That's almost a year ago. And the earliest genome out of USA, which was reported by the CDC, also coincides with this particular timeline. So it's sort of suggesting that the US genome was probably very close to the ancestor of the particular cluster. And over almost 1 year, it has been spreading, probably sustained human-to-human transmission, to sort of being detected today as a cluster of genomes which are completely unlinked from the European cluster or the B1 cluster. Now, of course, this was very important and interesting to us because this could suggest a different things. Of course, one of it is, why has been it transmitting for the last 1 year, probably undetected? Where has this been coming from? And more importantly, is it very different in between the other big cluster or the B1 cluster in terms of the rates of mutations? Now, what we know about this particular cluster, it's very characteristic. It has 16 genetic changes across the genome, and these are quite distinct or different from what we know for the B1 cluster. Just for comparison, I've listed out the mutations for both the clusters. So it is not like an error, but it is indeed a distinctly different cluster. And much recently, 2 days ago, there is another genome that has been deposited from the Thailand NIH, which also falls into this particular cluster. And even more surprisingly, you would, as you see here, there are at least three distinct clusters of A2, which sort of suggested that it indeed had been transmitting in a sustained manner through human-to-human contact for at least a year. And it had at some point of time differentiated into three different clusters, which has possibly different epidemiological origins, or also suggestive of different ways of transmitting. And of course, to put in context, this is the 2021 genome from the United States of America. Now, in terms of the genetic mutation rate, the B1 lineage has, as expected, a much larger mutation rate of 22 substitutions per year, approximately. And compared to the A2 lineage, which has pretty much half of that substitution rate, we still do not know why it has a much smaller substitution rate. One of the reasons could be that B1, since it's a large superspreader event, could undergo what we call as parallel evolution, where you have multiple different parallel human-to-human transmissions, could accelerate the rate of evolution of the pathogen. So this is possibly one of the reasons. The second reason, of course, is that the A2 cluster has a much smaller number of genomes, and therefore that could become an error in estimate. Doesn't look so because of the very sort of strict confidence margins in terms of the mutation rates. So in summary, we have genomes from three different clusters, three different countries, which comprise today the A2 lineage. So it's not from one single country, it's across the world from three distinct countries with apparently no epidemiological links between these three clusters. It is distinctly different from the B1 cluster, which is part of the European outbreak, the large European outbreak in 2022. It has very distinct set of genetic variations. Some of them are functional. We are actually going back and looking at it in much great detail. Probably a few weeks, we should be able to get some insights on that. And of course, a much earlier origin of the cluster, and a genome which traces back to 2021. We did confirm that on the date. It's not an error. It did come from 2021. Suggests a very cryptic and undetected spread of monkeypox, and typically suggestive of human-to-human transmission over almost 1 year. And to make it more curious, almost all cases, the two cases, at least in India and at least one case in Thailand, was related to travel to a foreign country. So that necessarily means that there are some other foreign countries which also has clusters of the A2 lineage, and probably more sequencing and more studies could probably unravel the full insight. Before I stop and take questions, of course, a lot of people to acknowledge the genomic data and deposits from multiple different countries and multiple different centers, including the CDC, the NIV in Pune, and the Thailand Red Cross and the NIH from Thailand. All the analysis was done by Bani, who is a graduate student at CSRI JB, and of course, the funding came from CSRI. Thank you very much.